This is the slideshow for Microbial Growth, Chapter 6, Part B. In the previous slideshow on Part A, we saw some of the selective and differential medias and other ways to be able to grow bacteria using complex medias like BHI or enriched agars like blood. Now we're going to talk a little bit more about how you actually obtain a pure culture. The only way that you can identify a bacteria is to only have one culture, one organism present. And so therefore, it's very important that a microbiologist is able to quickly obtain a pure culture. We define a pure culture by getting that from a single colony. From our microbial safari, we saw that a colony is a visible mass of bacteria where all of those cells came from one progenitor cell. So that way, everything within that colony is a clone of the original one. They're all the same. So in that way, we can transfer that to grow a pure culture, and that can be used for further testing and identification. So how do we get to that pure culture? Well, you're going to have a large number of bacteria in whatever sample you're starting with. So if you start to streak this out on a plate, the first point where you apply is going to be a loop full of bacteria, but that's going to have millions of cells. So you're going to get heavy confluent growth. But you want to be able to streak this out or dilute it. So what you're going to do is then flame your loop, and then you're going to take that cooled loop and drag it in and out of the previous quadrant. That is going to allow you to take some bacteria from that previous quadrant, but not a lot. And then as you streak that down the agar, you're actually going to separate or dilute the bacteria. You keep doing that again, applying the flame, then cool it, streak through the previous quadrant, streak down the agar, and most of the time we do what's referred to as a 4-quadrant streak. You have to get really good at this if you're working in a clinical microbiology lab, because you have to be able to obtain good isolated colonies as seen here on the blood agar plate. So a microbiologist is going to spend a lot of time streaking for isolation. Again, the technique is one where you are diluting or separating out the bacteria that are present. On the left-hand side, we see quadrant A being made from the original bacterial culture. And then to make quadrant B, we would have flamed the loop. We're going to let it cool, then go in and out of the previous culture. And then literally it's like scribbling on the agar, but pulling down and not going back over the previous lines to make quadrant B. We're then going to flame, do that again for quadrant C, and my quadrant D, as seen here, oftentimes usually ends up just being a little squiggle, but it's still a 4-quadrant streak. So if you allow that to incubate at 37 degrees Celsius, you see on the right-hand side a picture of a streak plate that has grown up so that we can now observe the colonies. If you look at the first streak area, you have heavy confluent growth. And if you look at this culture, it looks like everything is red there. However, as you start to see the bacteria in the second streak area and the third streak area, you start to see that you get fewer and fewer bacteria, and you're starting to see individual colonies. By the time you get out to that third streak area, you actually have individual isolated colonies. Once you've done that, you can observe there are 2 different bacteria on this plate. We couldn't tell that in the first streak, but when we have isolated colonies, we can actually see that there's specifically red and white colonies. So what you would want to do is pick a single white colony, a single red colony, transfer them to a new agar plate, do the streak technique again, and you would then have grown up a pure culture that could be used for further microbial identification or determining its antibiotic sensitivity, whatever is needed for that particular bacteria. So that's why doing a good streak plate is very important. It's a technique that takes time and practice. Unfortunately, we're just going to be doing it in the Labster this time, but it's still going to give you a feel for what the technique is about. So, how good is good? Could a pure culture of bacteria be obtained by a streak plate method if there were only one desired microbe in a bacterial suspension of billions? Probably not. That would be literally a 1 in a billion chance of trying to get that bacteria from that culture onto a plate. But in theory, yeah, if you actually picked it up in the loop, you could separate it out, but it would be virtually impossible, but it could happen. So when we are dealing with bacteria in the lab, we obviously want to protect ourselves because we're not always just growing up bacteria that could be just normal skin flora or various things. We could be working with potential pathogens. Therefore, we classify things by biosafety levels. So biosafety levels are going to be pathogen protection levels, which dictate a set of biocontainment precautions required to isolate dangerous biological agents in the laboratory setting. So if you have things that are a classification of BSL-1, no special precautions are required. You can just work with them on a benchtop or wherever. In a BSL-2 facility, which is what we would have in the teaching labs in PISB if we were actually in the lab this term, you would have to at all times have on a lab coat, gloves, and goggles for eye protection. BSL-3 is a biosafety cabinet, so you see the two individuals working in what people refer to as a biological hood. This is going to prevent airborne transmission because you have that window or that shield that pulls down in front of you and you put your hands in to be able to manipulate the culture. But these two individuals are actually working in a BSL-4 facility, the highest protected level, which is that you have a specialized room that has been sealed and actually has negative pressure so that you don't have currents taking things out as people go in and out. The air in that room is exhausted, and when you are using the hood, that hood is actually keeping the air flowing away from people, and that air gets exhausted twice or filtered twice as it is pulled away from those individuals. So it's very important, and you see these individuals have covering from head to toe. These are going to be highly pathogenic organisms. So if we look at our pyramid that correlates the biosafety levels and examples of microbes, we see our low-risk microbes in the BSL-1. These are things like Saccharomyces cerevisiae, the yeast that we use to make breads and some of our wines and things like E. coli. These are basically non-infectious bacteria. In the lab, even in teaching labs, we are considered a BSL-2 facility, and we have to wear personal protection because this includes things that could cause mild disease, but they aren't likely to be aerosolized, and so it's not likely that you're just going to inhale them to contract them. So things like hepatitis A, Streptococcus pyogenes, causative agent of strep throat, Borrelia burgdorferi, causative agent of Lyme's disease, Salmonella. These are all examples of organisms and viruses that you may have heard of that would be considered a BSL-2 level. BSL-3 microbes are either fairly exotic or they can cause potentially serious lethal disease, mostly through respiratory transmission. So it makes sense that things like Yersinia pestis, the causative agent of plague, Mycobacterium tuberculosis, the causative agent of TB, things like SARS, rabies, West Nile virus, hantavirus, these are all more problematic and so require BSL-3 level protection. When we talk about COVID-19, this would have to be handled in a BSL-3 facility if you were growing and propagating the virus. If you were just taking a sample and trying to identify if the virus was there from a sample, then you may be able to work in a BSL-2, but if you were definitely propagating and growing the virus, you would have to be in a BSL-3 facility. BSL-4 is only for the most dangerous high-risk microbes. These are ones that almost always cause disease and are frequently fatal and often without treatment or vaccines. So think of things like Ebola and smallpox, which obviously are very highly pathogenic and potentially deadly organisms. Although we're talking a lot about growing bacteria, we don't want to keep them growing all the time. That would take a lot of media, a lot of effort, because you have to keep giving them fresh media, keep transferring them over and over again. And the bacteria themselves may actually not like that and may not do well after you've passaged them a number of times. So therefore, we often want to preserve bacterial cultures so that we can store them and come back to them later. So one way would be to deep freeze them. You can put them in a solution and freeze them at -50 to -95 degrees Celsius. And surprisingly, that's just going to inhibit their growth, but they're not dead. We store all of our bacterial stocks in a -80 freezer so that when we need that new stock, we can just go there, pull it out, and start growing it back in either liquid culture or on a plate, and it does perfectly fine. We can also do lyophilization, which is freeze drying. You take a frozen culture at -54 to -72 degrees Celsius, but you add, in addition to that, vacuum to dehydrate. So you're going to suck all the water out of the cells, and then you end up with a powdered form of the bacterial culture. You can store this in a vial, seal it, and that way this can be shipped from one place to another or stored very easily so that when you want to work with it again, you take off the seal and simply inject in some liquid nutrient broth and the bacteria will start growing again. So preserving bacterial cultures are very important in making sure that we have the stocks available to be able to study or do whatever we need in the research and or clinical lab. So if the space station in Earth's orbit suddenly ruptured, humans obviously on board would die instantly from the cold and the vacuum. And would bacteria survive? Yep, because that would just simply be what we were doing in lyophilization. You're providing that freezing and that dehydration by vacuum. So the bacteria, they wouldn't be growing, but they certainly would not be killed if that were to occur. So bacteria are pretty amazing, not only from the way that they can withstand various things to try to kill them, but just also the rapid nature by which they grow. So we're going to spend some time talking about the growth of bacterial cultures. And on this slide, there's a link to a very short video that shows bacteria growing and how quickly you can start to have a very large population. So when bacteria reproduce, in general, prokaryotes divide by binary fission. So the bacteria that we're talking about that are clinically relevant organisms, these are going to be those that grow by binary fission, where they first just separate their genetic material, replicate it, and then literally are going to put down cell wall to make two new cells. Some bacteria can grow by what's referred to as budding. They can reproduce by just simply pinching off a daughter cell, as seen in the figure here. Others are a little bit mold-like, and these are referred to as actinomycetes. So they may produce reproductive spores. So they're kind of a bacteria, but they have fungal properties, as we'll talk about later when we look at our fungi in chapter 12. Other long filamentous bacteria may simply fragment or break off a viable piece, but by far, most bacteria are going to grow by binary fission. So binary fission, the first thing that happens is that the cell's going to start to elongate. The DNA is replicated and basically kind of gets partitioned to two ends or poles of the cell. As that happens, the cell wall and plasma membranes start to invaginate, and that is going to start dividing the cell into two daughter cells. That cross wall forms, and you start to see the peptidoglycan being laid down between the two new cells, and then eventually those separate. Remember, not everything separates, so sometimes things may stay in chains or clusters, as we saw in our bacterial arrangement in Chapter 4, but we're definitely making two new daughter cells. So in the figure here, we see a section of bacillus as it's starting to divide. You can see that partially formed cross wall as it's starting to invaginate, and that will become a cell wall that is complete so that two new daughter cells will then be able to separate. Cell division for bacteria happens exponentially. It grows very quickly in a doubling. So that means 1 becomes 2, 2 becomes 4, 4 becomes 8, and so on. So if we think about a visual representation of the numbers, we can look at the ball model there, where again, you start to see how quickly the replication occurs. But we can also think about that this can be expressed as a power of 2. And so that's an interesting and useful way to think about cell division for the prokaryotes. When we try to plot microbial growth, we have some issues. Yeah, when we think about them growing as that doubling, it's growing very quickly. But if we were just trying to count that on an ordinal scale, we would see that we wouldn't be able to tell the difference between some of the numbers, because unless we had drastic differences between them, it might not be so obvious on an ordinal scale. So if we look at the graph there, normally if you're just counting on an ordinary scale, you would have a J-shaped curve. And the problem with those J-shaped curves is that if you look at the number of bacteria at, for example, 10 generations, there's still over 1,000 bacteria. Some bacteria may be infectious with less than 10 bacteria, so having it look flatlined there is problematic. It isn't until you start to see large numbers that you really start to see that upslope of that J-shaped curve. So that would be problematic if we're trying to represent this and work with any type of predictive value for the curve. So instead, we plot bacterial growth on a log scale. So the left-hand scale shows the log number of cells, and these are going to be logs of 10. And so this is going to give us a nice straight line when plotted. So we see that dashed line is going to be a much easier way to be able to use predictive ability. So you could, if you have that nice plotted linear line and you know the number of generations you let a bacteria grow, you could then work backwards and figure out the number of cells. So plotting things on a log graph is going to be much more useful than trying to just count the number of cells and use that J-shaped curve. The slide here has a video that is going to look at not only the binary fission, but also bacterial growth and the curve that is generated as we go through the different phases. So we're going to talk about that in more detail in the following slides. So bacteria double. Therefore, when we talk about the generation time, many people also refer to that as the doubling time, the time required for the cell to divide or the population to double in number. Most bacteria have a generation time of 1 to 3 hours. E. coli is one of the fastest growing organisms. It has a replication time. It doubles every 20 minutes. That's why E. coli has become one of the workhorses of molecular biology. It is easy to grow and it grows very quickly. Staph aureus isn't too far behind at a generation time of 30 minutes. So again, that's problematic for staph, like when it causes staphylococcal food poisoning. The fact that it can grow so quickly and then produce enough toxin if it's left in that temperature abuse, that danger zone. So that's why we start to see why looking at how quickly organisms can grow is also part of understanding some of the diseases that they can cause. Mycobacterium tuberculosis, on the other hand, the causative agent of TB, is one of the slowest growing organisms, taking over 13 hours to be able to double. This also becomes problematic as people become non-compliant with their antibiotic therapy. The slow-growing bacteria, you need to make sure that you're taking all of your treatment so that you kill the organism and not just say, oh, starting to feel better, because the bacteria is still there, just growing slowly. We often want to calculate the generation time. So counting bacteria can be kind of difficult, but we're going to have to do a little bit of math here. So to calculate the final number of cells in a culture, you would take the initial number of cells that you've counted and multiply that by 2 raised to the number of generations to get the final count. For example, if we had 5 cells and allowed them to divide 9 times, then to calculate the final number, we would take 5, the original number of cells, times 2, because again, everything doubles, and that's raised to the number of generations, which in this case is 9. So we would end up with a population of 2,560 cells. So that's useful, but oftentimes what we want to report is what's referred to as that generation time. When we report generation times, it's the number of minutes per generation. So to calculate that number of generations, we have to go back to doing things in log. So we must convert everything to a standard log, which is based on 10, in order to make sense of these numbers. The log of 2 is equal to 0.301 because one cell divides into 2, so that's going to figure prominently into our equation. So to calculate the number of generations, we're going to take the log number of cells at the end and subtract from that the log number of cells at the beginning. That is then put over the log of 2, which is the 0.301. So in order to get it into the generation time, you're going to take the number of generations that you just calculated and use that in the denominator to convert things into minutes once you know how many hours you let that culture grow. So generation time is going to be some number as minutes per generation. So in this example, if we take 100 cells and allow them to grow for 5 hours, we see we get a lot of cells. So we get over 1 million cells, and we need both of those numbers to plug into our equation. So first, again, we're going to calculate the number of generations. So we're going to take that log number of over 1 million cells there. So we have 1.7 plus million cells. So the log of that number at the end. Subtract from that the log number of the bacteria at the beginning. So the log number of 100. And that is over the denominator, the log of 2, which is the 0.301. That is going to give us the number of generations. That goes in our denominator. And if you actually do that calculation and convert that into the minutes per generation, that calculates out to be 21 minutes per generation. Most likely this is a E. coli because not too many other things are going to replicate that quickly and have a generation time of around 20 minutes per generation. So why would we want to do this? Well, oftentimes we're trying to figure out if something worked, a preservative, an antibiotic, or something. So in the example here, we're going to look to see if a preservative actually inhibited the growth of the bacteria from the previous slide. So let's suppose that we have 900 of the same bacteria from that previous slide, and we're growing them under the same conditions, but now we're going to add a preservative to the culture. We're going to let it grow for 15 hours, and when we do so, we count the cells, and we see this time we have over 3.2 million cells. So we have to take the end log, so the log of that over 3.2 million cells, figure that out, subtract from that the log of 900, which is what was the starting number of bacteria, and put that over the log of 2, which is the 0.301. When you do that calculation, you end up with 11.8 generations. That number of generations then goes into the denominator, and we want to convert things to our minutes per generation, and we know that we let the culture grow up for 15 hours, so we do that calculation, and we see that the generation time with the preservative is now 76.3 minutes per generation. So did the preservative inhibit growth? Greater than 76 minutes per generation compared to the 21 minutes per generation without the preservative says yes, the preservative did inhibit. So the beauty of doing these types of calculations is that I don't have to have the same number of bacteria to start with each time. That would be very difficult. And I don't have to always have the same number of hours. So it doesn't matter if I let the previous one go for 10 hours and this one is at 15 hours or whatever the case may be. It's all going to be factored in there, and that way I can compare two populations, in this case one with the preservative and one without, and truly identify by looking, did it take longer for the bacteria to double? If it did, then yeah, probably that preservative did indeed work. So what we've looked at so far is just simply the exponential, or what's referred to as the log growth. That was really just a small portion of what's the bacterial growth curve of bacteria. The growth curve, if you look at the population over time, we see that everything is going to start out in the lag phase. There's the flat line because there's little or no cell division, but don't be fooled, the cell could actually be very metabolically active. It has to be replicating its DNA. It maybe needs to make enzymes or structural proteins to be able to divide the cell. So there's no cell division, so therefore the line is flat. However, it still is going to be metabolically active. When it goes into the log phase, that's when we start to see the cells are actively reproducing. And that's when you're going to have that logarithmic or exponential growth as the curve shoots up. This is going to mean that the bacteria is at its growth maximum. So it's at its optimum, it's growing as quickly as it can. But think about the terminology. The generation time is at its minimum. It's the least amount of time that it takes for that bacteria to double. And it's going to then reach that minimum generation time and keep doing that as it goes through that logarithmic plot. So that gives us that nice straight ascending line that we were referring to before. These are the most metabolically active cells, and therefore they're also the most susceptible to adverse conditions. So if you want to kill a culture of bacteria, it should be actively growing. So that's when you want to treat it with that preservative or that antibiotic or that disinfectant. You want to have something that is actively growing to be able to incorporate or utilize those types of adverse conditions or chemicals or antibiotics, whatever the case may be. At some point, the culture is going to reach a stationary phase. The number of cells that are being produced are going to equal the number of cells that are actually dying. So you have a flattened portion. This can also be that maybe nutrients are running out, various things that are causing the cells just not to be able to actively replicate as quickly as what they were doing before. Eventually, the culture is going to go through the death phase. The number of deaths exceeds the number of new cells. The culture starts dying out. Maybe the pH has changed too much. Maybe nutrients are running out. but it is going to have it's not going to be able to just carry on forever. If you wanted to keep it growing, you would then have to transfer it to a new beaker of broth or something, a new flask, to be able to provide it with new nutrients. Understanding the bacterial growth curve is very important, not only from the standpoint of, say, for example, in making various food products. If you wanted to make those wines and cheeses and various things by fermentation, you would have to get your culture into log phase. So those Saccharomyces aren't going to produce the ethanol that you need unless you get them growing, and you want to then harvest in that log phase the products that they're making. From the standpoint of infectious diseases, again, log phase is when you want to be treating. That's when you want to give the antibiotic, because the lag phase, you wouldn't know that you have the infection. Think of it when you get exposed to something. It's dependent on the organism, but there's always a certain incubation period before you start seeing active signs and symptoms of that disease. Those active signs and symptoms of the disease are occurring as the bacteria are growing in that log phase. And so therefore, that's when you want to treat it. That's when you want to actually be doing something to try to inhibit it if you're talking about an infectious agent. Hopefully, that eventually puts things into that stationary phase where you've turned the corner and you start to level off and eventually clear the infection. So bacterial populations, because they follow this sequential series of growth, lag to log, stationary, and then death, it's critical for our understanding of the population dynamics to be able to control bacterial infections, use them for food preservation or in making our desired food products. So it's very important to understand the bacterial growth curve. Most other organisms can't do exponential growth the way that bacteria can. So the question here of if two mice loved each other and started a family, would they be able to make anything comparable to a bacterial growth curve? No. They're going to have to go through sexual reproduction, which just isn't going to be able to reach the level of exponential growth that we see with bacteria. So bacterial growth curves are unique, and that's why it's so important in understanding microbial populations, both for treatment or when we want to grow them, in order to be able to appropriately either kill them or give them what they need to be able to grow exponentially. So if we're growing these bacteria and we said we had a certain number of cells, obviously we need to count them. The microbiologist spends a lot of time trying to figure out how many bacteria are there. This can be both by direct and indirect methods, and we're going to take a look at those various methods in the subsequent slides. So one way to count bacteria is literally to count them directly underneath a microscope. Bacteria are very small, so you're looking at them under oil immersion, and that means that it's really hard to be able to keep track of them. So we actually have these little slides and cover slip that allow us to put a certain volume underneath a cover glass that has a grid on it. So if you're doing a microscopic count, you're going to have these squares that are going to help you keep track of things. So for the microscopic count, you're going to count the cells that are in this large square. The large square in the picture there is outlined in blue, and you can see that it has 16 squares, 4 across and 4 down. Those smaller squares are going to help you keep your place as you're counting the number that's in the large square that's shown there. If you look at that outlined blue square and count the bacterial cells, you'll see that there's 14 bacterial cells there. So not only is the grid helping you count, we also know the volume of fluid that's underneath that large square. So that is measured as 1 over 1.25 millionth of a milliliter. And you can then do the calculation of taking the number of cells and multiplying that by the volume. Doing numbers that are up in the millions is problematic. So oftentimes we put things in scientific notation. So we could also say that this is 1.75 times 10 to the 7th cells per milliliter. And those are much easier to say and also just to keep track of in general. If you don't remember your scientific notation, here's a little cheat sheet of what you do. So in example 1 there, we have 52,314. And if we want to put that into scientific notation, we're going to move the decimal 4 places to the left, and that would be 5.2314 times 10 to the 4th. So we see the examples there, and putting things in scientific notation just usually is easier in trying to keep track of these very large numbers. Besides counting things directly underneath the microscope, we can also do a direct plate count. After we spread bacteria on a plate, we can incubate them and then literally count the colonies. For a microbiologist, a countable number to give us a valid count is going to be between 30 and 300 colonies, or what are referred to as colony-forming units, or CFUs. We're not going to just get 30 to 300 colonies unless we do something to dilute things out. Even when we did the streak plate, if we just put a loop full of bacteria on that first quadrant, we have way too many cells to be able to count. So we do what is referred to as a serial dilution, where we keep diluting the bacteria over and over again. In order to do a serial dilution, we're going to take an original inoculum and take a small volume and put that into a larger volume to dilute. Typically, we do a 1 to 10 dilution. So a 1 to 10 dilution, the 1 is the amount transferred. In this case, you can see from the pink arrows that we're transferring 1 ml over the total volume. We have a test tube that has 9 ml of broth in it, but once we've transferred in the 1 ml, we now have a total volume of 10 ml in that tube, and so that's going to give us our 1 to 10 dilution. We're going to do that over again, so we're going to mix up that 1 to 10 dilution and then take out of there 1 ml and transfer it to a new tube of 9 ml of broth. That would be a 1 to 100 dilution. 1 to 10 dilution times another 1 to 10 dilution gives us a 1 to 100. And we keep doing that over and over again. In this case, we have done 5 1 to 10 dilutions. So that would be like taking 1 to 10, multiplying each of those out, so that 1 to 10 times 1 to 10 times 1 to 10 times 1 to 10 times 1 to 10, 5 times, till we get the 1 to 100,000. So now we have the tubes diluted. We can see that visually by the color keeps getting lighter and lighter. That's representing that there's fewer bacteria each time we do a dilution. But now we want to count it. So we're going to take 1 ml and put that onto a plate and spread it out. If we look at the 1 to 10 dilution, that's going to be way too many colonies to be able to count. And so that's why it's labeled as TNC, too numerous to count. Even looking at the 1 to 100, it's maybe fewer colonies, but it's still too numerous to count. And even when we get to the 1 to 1,000, there's clumps of bacteria. It's still not a countable number, so it's still listed as TNC. The one that we want to use to give us a statistically valid countable number is going to be the 1 to 10,000. That is starred there because when we look at that plate, it has that magic number, the 30 to 300 colonies. If you count that plate, you see that it actually has 32 colonies there. You can continue on to the next plate for the 1 to 100,000. And yes, there are 4 colonies on there, but that's too few colonies to be statistically valid. Things could just be sticking to the pipette, whatever, and so we want to focus on the plate that has between 30 to 300 colonies to be able to do our calculation. So our calculation, we take the number of colonies on that plate and multiply that by the reciprocal of the dilution of the sample. Since it was a 1 to 10,000, we're going to take those 32 colonies that are on the plate and then multiply that times the 10,000. So that is going to give us what is referred to as the titer, and in this case we see that that's 320,000 colony-forming units per mil in the original sample. Or the easier way to do that is to say that putting it in scientific notation, that is 3.2 times 10 to the 5th colony-forming units per mil. You start with the decimal point and you would move it 5 places until you have the decimal point between the 3 and the 2. If you're confused, go back to the previous slide and review your scientific notation. So in this way, we can actually see how many bacteria, or count how many bacteria are in that original inoculum or that original flask, and that is reported as the titer in colony-forming units per mil. How do we get the bacteria spread out? Well, there's a couple of different methods. The pour plate method isn't all that useful. You could actually mix the bacteria with melted agar that has been slightly cooled and then swirl that in the plate. But the problem with that is that you get colonies growing on the agar and colonies growing in the agar. It's hard to count them, and plus if you are interested in one that's in the agar, it's just hard to get it to be able to streak it out onto a new plate. So more typically you do the spread plate method. You take a volume of bacteria, put it on the plate, use what people refer to as a hockey stick to kind of spread it out, and then incubate it overnight. This is then going to give you colonies that grow only on the surface, so not only can you count the colonies, but they're easily isolated if you want to propagate those colonies further for identification or microbial sensitivity to an antibiotic or disinfectant or whatever the case may be. Filtration is another way that you can actually count bacteria. So a filter just has pores in it that are going to allow liquid to go through, but doesn't allow the bacteria itself to go through. So if you have a larger volume, you can let the water go through and the bacteria are going to be trapped on the filter. You can take that filter and put it on an agar plate to provide it with nutrients, and the bacterial colonies are going to grow, and similar to what you did before, you would just count those colonies and you would know what volume of liquid that you put through so that you could figure out the titer. We also have another direct method that's referred to as the most probable number, or MPN. This is very useful when you're trying to figure out if maybe a natural body of water, a lake, a pond, or something is contaminated with fecal organisms. So this is going to be based on testing for what are referred to as coliform bacteria, those gram-negative rods that are lactose positive that we talked about with the MacConkeys and EMB, things like E. coli and Enterobacter. They're going to indicate that things may be fecally contaminated. If you have a high coliform number, that would indicate that the waters are fecally contaminated, and the chance or the probability that a pathogen could be present is high, so those waters would be deemed unsafe. It would be really hard to try to find the pathogen. The pathogen is probably in low numbers, and so therefore it'd be like looking for the needle in the haystack. So doing the most probable number based on the normal fecal flora is the best way to be able to get that coliform count to determine whether there's too much fecal contamination. So if you have a volume of water, lake water or whatever, you're going to inoculate a set of 5 tubes with different volumes of that source. So 10 mils are going to go into 5 tubes of lactose broth, 1 mil and then another set of tubes gets 0.1 mil. You're then going to let them grow and look for those that are able to ferment the lactose. In this case, this is going to be indicated by the darker color. So if you look at the 5 tubes that were inoculated with the 10 mils of our water sample, we see we have 5 of the tubes are positive. So all the tubes are positive for the lactose-positive coliform bacteria. When we look at the tubes that have been inoculated with the 1 mil, only 3 out of the 5 tubes are positive. And for the 0.1 mil, only 1 tube is going to be positive in that series of 5. So that's going to give us what's referred to as the combination of positives. And that can then be put into a chart that's based on previous counts. So the combination of positives that we've generated is 531. So if we look at our MPN table, we see that the MPN index per 100 mils of water is 110. The numbers are based on counts that were actually done. So we can generate a 95% confidence limit that says that we believe the real number is somewhere between the lower limit of 34 and the upper limit could be as high as 250, but it's getting us in that ballpark. Now, this may not seem like it's a direct countable number, but because it's based on counts that have been previously done, this is a direct method and one that allows us to, again, quickly determine whether waters are fecally contaminated, and we wouldn't want people to swim or fish or do recreational boating possibly on those waters at that time. That becomes very important during the summer months, as warmer temperatures means those coliform bacteria are going to tend to proliferate. If there's fecal contamination, again, we're not certain that there's a pathogen there, but the probability is high, so the waters would be deemed unsafe. So those were the direct methods, but we can also do indirect methods that give us an estimate of bacterial numbers. We can do things like metabolic activity. So this can be based on enzymatic activity. If you have the same bacteria and you grow them under different conditions, you can compare the enzymatic activity for a specific enzyme, say for example lipase. And if you have a higher lipase activity in one population versus the other, then you can say that the higher enzymatic activity is associated with a higher number of bacteria. But you have to be comparing the same bacteria. So you can't be comparing apples to oranges, so to speak. So it's indirect, but it's going to give you an estimate of the number of bacteria if the enzymatic activity is higher. You can also do things by dry weight. Just simply suck all the water out of the bacteria, dehydrate them, and weigh them on a filter paper. The one that weighs more, obviously there was more bacteria there. So metabolic activity is used quite frequently. Dry weight, not so much. But a lot of times we use turbidity using a spec. So turbidity is how cloudy is that solution or that culture of bacteria based on the number of cells that are there. So you use a spectrophotometer that's going to be able to detect the light going through the suspension. If you have a light source and you have a blank tube where nothing is growing, the light's going to be able to go directly through to the light detector, and you would have 100% transmittance. However, if you have a bacterial culture, as the light tries to go through, it's going to hit the bacteria and scatter. So very little light is actually going to go through the culture to be detected by the light-sensitive detector. Therefore, the percent transmittance is going to go down. The more bacteria, the lower the percent transmittance, and this way this can be correlated with the amount of bacteria within that culture. So measuring microbial growth is very important. We do it all the time to be able to understand the infectious process or the disinfection process. And so direct methods such as Plate counts, filtration, the most probable number, and directly counting under the microscope are all examples of how we get real numbers by direct methods. Indirect methods are turbidity in a speck, metabolic activity using enzyme activity, dry weight, literally measuring the mass when we dry the bacteria on filter paper or something. Those are all indirect. They allow us comparisons, but not absolute numbers. So this concludes the chapter on growth. We're going to, in the next chapter, look at how we control the growth, because even though it's great fun to look at the microbial growth, most of the time we're trying to control it from a clinical standpoint. So the microbiologists, we want to grow and figure all that out, but from clinically relevant work, we're probably more interested in making sure that we're controlling that growth and disinfecting when necessary and not allowing microbial growth to cause infection. So we'll look at that on the next lecture as we evaluate Chapter 7 and talk about the control of microbial growth.